Hi. I want to thank Tao and Fred and Francis for hosting and inviting us here. It's been a really fun time in Cranfield getting to see all that you're building here as part of EBIC. So I'm an assistant professor at Princeton University. I just started my group just over a year ago. So we're just starting getting off the ground. And so I'm jointly appointed in the Civil Environmental Engineering Department, but also the Oman Darling Bioengineering Institute. And so I'm really a bioengineer by training, but applying bioengineering to sort of environmental challenges. And so today what I was going to tell you about is a little bit of the work that we're doing on engineering microbes as what we like to call living electronics that we use for sensors to build sensors for environmental contaminants in particular. So as we build the infrastructure of the future, we really want this water infrastructure to be adaptable to changing conditions, right? We have changing climate. We might want to adapt these water systems to deal with different flow rates that we might get from storm flows or from perturbations that might happen from things like human activity. We might introduce a chemical that we don't want into our water system and be able to detect this and, modify this process on the fly and make this adaptable. I don't think I have to convince the folks here that it'd be great to be deploying, advanced environmental biotechnologies to do some of these challenges, but really, how do we make the decisions on when to start up some of these processes in these reactors, right? And so we have really great sensors for things like flow and pH and turbidity that can already be deployed in these settings to build systems like you see on this slide here. But one thing that's rather challenging is to detect molecules like organic chemicals that we might want to detect or biological molecules that are hazardous. So things like nucleic acids from antimicrobial resistance genes or from viral particles. And how do we actually go about digitizing this molecular information in situ in these types of settings where we want to sense this, rather than having to take a sample, go back to an analytical lab, do a measurement, make this decision, after some amount of time. And so this is one of the focuses of my group, is really trying to tackle this molecular digitalization question here, and if we can build sensors for that. And the way that we tackle this problem is that we try to use synthetic biology or engineering biology to overcome this problem, really to program bacteria to convert chemical information directly into electrical signals. And so the idea here is we want to take advantage of the ability that biology has for sensing the world around it. Cells have evolved quite unique strategies for measuring the molecules that are around them so they can make decisions on when they want to grow in different places. If you look at bacterial genomes, you look at the proteome content of them, up to 2% of that proteome is devoted just to sensor proteins, molecules just to detect their environment. And so we want to take advantage of some of that, evolutionary diversity that microbes have for sensing their environment and build devices out of them. And so the other end of this is thinking about what is the signal that these cells are actually producing, right? So most people who think about biosensors, you might think about making something like a visual output, like a fluorescent protein or luminescence, right? These work wonderfully on paper-based sensors that folks here are building, or things where you want to do point-of-use diagnostics, but they don't work so well out in environmental samples. So if you want to go out and put this in a river or in a wastewater treatment facility, these are very opaque environments. You can't see what you're trying to sense here. And so what we're trying to do is interface cells directly with electrodes so they can generate electrical signals that can work in sort of less transparent environments that are more challenging, with the idea of ultimately packaging these all together into devices that you can put out into your waterways to detect when a chemical hazard appears in this environment. So really trying to build deployable sensor networks out of these biological devices that we're building. And so people who think about biosensors, we heard a really great example in the very talk right before this, would typically go to promoters and controlling gene expression, the production of a particular molecule. This works wonderfully. It's very modular. You can pick a promoter from many different organisms and build a circuit to do this. But there's some challenges in the speed at which these respond, right? So you have to transcribe a gene, translate a gene. This takes, at the fastest, a half an hour to respond to something. It's also very costly for the cell, huge amounts of ATP to produce proteins that you can actually measure, and for these proteins to come and mature inside of the cell. And so we take a slightly different approach in how we build biosensors. So rather than trying to control transcription and translation, we try to control the flow of energy and mass through metabolism and use this as sort of our way of controlling in building these types of sensors. And the other reason why we do that is because metabolism is inherently electrical in nature. So this idea of trying to want to make electrical signals at the end of the day, metabolism is a great place to start if you're thinking about that. And so this is an abstraction of the metabolism that's happening in all of your mitochondria right now as you're digesting the food we just ate. If you're an electrical engineer, you might know what this circuit is. This is what we would call a DC-DC power converter. takes in electrons at one potential and it modulates them up to another potential. And that's what's happening in all of our cells. We strip these low potential electrons off of organics like glucose. We hand them off to different redox carriers that have successively increasing potentials until we dump them off on an electron sink. And so we wanted to see if we could take this electrical circuit that operates in every cell on the planet and use this to build sensors out of. But right now we have no way of actually interfacing this with the device, right? Our terminal electron acceptor in this case is oxygen. And so we really needed to come up with what is the electronic interface that you can use to have cells directly produce electricity on electrode surfaces, right? And fortunately, there's a suite of microbes who've already evolved to do this out in the environment. We've heard some of them mentioned earlier in the talks. So there's a subsection of microbiology we call electromicrobiology that's devoted to looking at these types of organisms. You have organisms like Geobacter, which is a very famous example, or Shigella, another very famous example. And they've evolved these molecular machinery here that you see in red to move electrons out past the outer membrane of these cells. So these bacteria, they have a lipid layer on their outer surface or a protein layer on their outer surface, and it functions as an insulator typically. You wouldn't be able to pass electrons across this. And so they've come up with these proteins that allow electrons to transmit across them, and then these things are embedded and pass through these membranes. And so we take advantage of some of these features that biology's evolved to try to interface cells directly with electrodes that we're building. And the way that we go about actually manipulating this is using those tools of synthetic biology to program how these types of organisms behave. So really our ability to take and program DNA and RNA and proteins at sort of the sequence level and using these to program these particular cells to perform these different types of functions. And so today I'm just going to talk about our work on doing sensing, but we also do some work thinking about how you can have cells compute information about their environment or catalyze reactions, maybe in the context of bioremediation. And so one of the model organisms we like to work on in my group is Shigella onatensis. These are these cells here that you see in green, living on an iron oxide particle. And so these are facultative aerobic organisms. They live in the bottoms of lakes. If you go and look in the lake's sediments, they live in sort of the upper layer of the sediment. They love to breathe oxygen just like we do, but they also have the ability to respire off of many different materials, including this iron oxide that they're living on here. And so these cells, they're not just living on this particle, they're actually using this particle for their metabolism to produce energy. And so they use some of those proteins I showed you on that previous slide to actually move electrons out to this rock that they're living on. And so we take advantage of their ability to do this to interact and interface them with electrodes. And so this image that I'm showing you here on the right of the screen is an indium tin oxide electrode, so this transparent electrode material on the right side, and then just glass as an insulator on the left here. And what you see is as we change the potential of this electrode, these cells are actually coupling their metabolism to this electrode. They're generating proton motive force. That's what you're actually seeing with the green color. We're using these Nernstian dyes here. And so they're able to only transport electrons when those electrodes are poised at these favorable potentials. And the reason they're able to do this is because they've evolved these strategies for moving electrons at many different scales. And so at the smallest scale, the key innovation that they have in their genome are these multi-heme cytochromes, this particular complex MTRCAB. So it's 3 proteins here. It's this beta barrel that's in the membrane, and then a multi-heme cytochrome that threads through that barrel, and then a multi-heme cytochrome that's out on the cell surface. And this allows these organisms to move those electrons across that nanometer distant barrier that is the bilayer on the outside of the cell. But these cytochromes are not just static inside of the cells, they are moving around in the fluid membrane, so they can actually diffuse and move. And so this allows these organisms to actually move electrons at quite a longer distance in this case. And so each of these red traces that you're seeing here is actually one of these cytochromes that's been labeled with a quantum dot. You can see it actually migrating across a membrane tubule that connects these 2 cells. So they can move electrons at sort of these micron distances on the order of the size of the cell. But these cells, they also don't live in isolation in their environment. They live in the context of biofilms typically. And so they can move these electrons even further through these biofilms. So they have intercellular electron transfer that happens between cells that are next to one another within this biofilm. And so all of this red matter that you see on these 2 opaque electrodes in the background are these biofilms. And the electrons can flow from one electrode to the other through that biofilm. And on this case, it's on the order of about 5 cell lengths that these electrons are able to move. And so these cells, they typically grow like this very sort of amorphous smattering of cells. They're not something you want to build an electrical circuit out of. But really what we wanted to see is if we could take this material and turn it into something we can program and actually treat it somewhat how we fabricate semiconductors and things like that. So could we take something that looks like this and turn it into something that looks like this, more like an electrical circuit that we could think about bridging electrodes and measuring current flow through these electrodes. And the way that we go about doing this is using, again, these tools from synthetic biology. So we use optogenetics to control how these biofilms form on surfaces. And so the circuit, what this is describing to you is basically blue light induced gene expression of these two proteins here, CDRA and CDRB. These are biofilm formation proteins from Pseudomonas aeruginosa. But if you express these in Shewanella, it makes these cells very sticky. They like to clump together. They also like to adhere to surfaces that they're around in their environment. And so by hooking this up to blue light, what you can do is use typical lithography approaches where we use a photomask and a very simple LED light source here so that we can pattern these biofilms in different shapes onto the surface of the electrodes that we're working with. And so we typically work with those indium tin oxide electrodes that I mentioned earlier. And so after we got this all pushed into the cell, the very first thing we decided to do was to print our funding agency's name to make sure that they were happy. And so this is funded by the Office of Navy Research in the US. And so they were happy to see that we could spell out their name on here. But what we typically do is print rather boring shapes, things like rectangles. And the reason why we like to do this is because rectangles are really easy to do math on. And so part of our goal here was to really measure the intrinsic conductivity of these biofilms, treating them like a material and thinking about how you can build larger circuits out of these. But we first needed to have an understanding of the geometry that you're passing current through. And so we can make rectangles of different shapes. We can make many different rectangles on a particular surface, all with this tool here, which is nice. And so what we typically go about doing then is to take these and print them onto what we call interdigitated electrodes, which you can sort of see here. What you have is a source electrode and a drain electrode that are interdigitated like that. And so we pass current from the source to the drain through the biofilm. That's really what we're doing here. And so seeing a top-down view and then a side view with the cells sort of layered on top in red. And then this is an image of what this actually looks like in the lab. And so because these electrodes are transparent, this is actually a schematic of what the electrode looks like. And then this blue or purple area that you're seeing here is the actual biofilm on this electrode. And so we pattern it over that rather large interdigitated region of these electrodes. And then what we do is we perform some electrochemistry on these biofilms. And so we'll pass current at different voltages and look at how this flows through that biofilm. I'm not going to go through the cyclic voltammetry and all the details of that. This is a product of multiple measurements at the same time that we're doing. But when we take these two electrodes and we keep them at the same potential relative to one another, so there's no driving force between them, you get this very flat conduction current. There's no electrons that are really flowing through this biofilm. If you were to look at the CVs, you would see electrons flowing between the biofilm and the electrodes, but none between those two electrodes. But now if we take these two electrodes and we apply source drain voltages, you get a very different profile on what this conduction current looks like. And so as we start from sort of this low potential to this high potential, you can see these peak-like features that appear here centered around this one particular redox potential. And so that maps to the redox potential of these multi-heme cytochromes. This is where they actually are favorable to take up and donate electrons. And so you see this sort of redox conduction. This looks a lot like redox polymers if you make electronics out of those, but made out of living cells that we pattern on top of the electrode. And so if you take this peak feature and you look at sort of the magnitude of the conduction current that you get here, you can plot this versus the different voltages that we're applying across the two electrodes. So the source drain voltage goes from 0 to 60 millivolts here. And if you look at sort of the magnitude of that current, you get this nice linear relationship. And you can take this and go about calculating what the conductivity of this biofilm is. And so this is basically just Ohm's law. It's nothing particularly complicated. And then a bit of the geometry of the biofilm. So we make a confocal measurement of the thickness. We know what the outer, the X and the Y of this particular biofilm is. And with that, we can derive out this sigma here, this conductivity of the biofilm. And so this is just a wild type Shewanella biofilm with that optogenetic circuit. It's about 4.5 nanosiemens per centimeter. If you know conductive materials, it's not the most conductive thing in the world, but it's enough to measure, which is all we care about in the sensor world in this case. We're not trying to pass loads of current through this thing. And so this was sort of the first measurement of the intrinsic conductivity of these biofilms, but we wanted to see if we could take this a step further and tune the conductivity in these biofilms and see if we couldn't do this in a chemical dependent manner. And so use tunable conductivity as sort of an output for a sensor modality, for example. And so we take those same cells, that Shewanella organism that I'm talking about, and we go into its genome and we delete all of the cytochromes from that genome. So the cell is no longer producing these molecules that allow electrons to be transported. You get a profile that looks very similar to this. It's relatively flat, even though in this case we are applying a source drain voltage of about 20 millivolts. This isn't in the absence of that voltage. And then what we did is we added a rather simple gene circuit here to control the concentration of these cytochromes that these cells are making. And so these cells, in the presence of vanillic acid, will begin to produce this MTRC A and B proteins, and then they'll begin to make cytochromes. And so then what we did is we went ahead and made all these measurements. at different concentrations of vanillic acid. And as you can see here, we get that phenotype coming right back, where they look a lot like the wild type here, and it's in a chemical dependent manner as far as the magnitude of the currents that we're getting out. And so what we can end up doing here is look at sort of, the Hill function or the transfer function, if you were thinking about this as someone who does a lot of gene circuits for the output of conductivity in this biofilm rather than fluorescence. And so we can tune this between about 0 to 6 nanosiemens per centimeter. We didn't, increase the amount of conductivity that these have by much. You can do just a little bit. They're more or less maxed out in their wild type. And we have this response curve that's really defined by the transcription factor that binds vanillic acid. So it responds to about 6 micromolar of this vanilla molecule. This isn't necessarily a molecule we would want to sense for any real purpose. This is literally vanilla that you can go to your grocery store. So if you want to know that, you can see that here. But we can adapt this to bind effectively any other transcription factor. So we could take something that binds PFAS if we had a nice transcription factor and make this respond to that, or zinc or lead or any of these well-known pairs of promoters and transcription factors that exist. But like I said at the beginning, these processes are rather slow. So what I'm not showing you here is that it takes 16 hours for these cells to grow on this biofilm in the presence of this molecule. That's not a very fast sensor, right? It's quite slow. And so we wanted to next see if we could really speed this process up. Could we build sensors that actually have very quick time responses? And so if we come back to that electrical circuit that I was showing you at the beginning here, we have this nice electronic interface that works well for getting cells to produce current, but we need a way to actually control the current that flows through this particular system. So can we introduce an element within the circuit that is actually gateable and that can function like a switch to control the flow of electrons? And so what we did here was to turn to other sets of redox proteins that cells can make. And so this is a rather comprehensive list of all of the protein electron carriers that biology has evolved. We have very low potential electron carriers like ferredoxins that you find in photosynthesis and a lot of anaerobic organisms. You have very high potential things like rusty cyan and blue copper proteins that are important in respiratory processes. And cells use these for different types of chemistry. That's why they have all of these different potentials, and these all have different cofactors in them. Some bind just inorganic molecules, some like the cytochromes bind hybrid organic, inorganic cofactors. And so they're all used for different types of purposes. And so we wanted to see, they do one particular function. They take an electron from one enzyme and they give it to another enzyme. So they're not really dynamic. They don't respond to molecules. And so we wanted to see if we could take one of these proteins and make it allosteric, as we would call it in sort of the enzymology world. And so we specifically took some of those 2-iron 2-sulfur ferredoxins and asked the question, can we make this into a switch that responds to a chemical? And so basically in the absence of that chemical, you have no electron transfer within this circuit that we want. And then when you add this ligand that binds this red ligand binding domain, you can turn on electron transfer within this circuit. This was sort of the main idea, is can we engineer these proteins to have this type of function? And so the way that we go about actually building new types of switches like this is to use the tools of protein engineering. A lot of what my lab does is to leverage the approaches of directed evolution, so coupling the function of a particular protein to a way of selecting for more biomass in that way. So this could be producing a colored molecule that you can do FACS sorting on, or coupling it to the growth of the cells. And so for this example, what we did is we used an oxotrophic E. coli strain that couldn't make sulfide, and then we added in a pathway that was dependent on this electron transfer event to make sulfide. And so these cells would only grow if electrons were flowing within this metabolic pathway here. And then we use that as a tool to screen libraries of mutant versions of these proteins. So we'll take that ferredoxin and we'll insert ligand binding domains at every possible site within that particular protein sequence, and then look for which of these proteins are active after we have done that mutation to them. And so we do this by using sort of deep sequencing to actually assess which variants are enriched within a population. And so we'll grow these cells in a selective and a non-selective condition. We'll extract their DNA. We'll compare the number of each of the variants within each of these conditions. And then we make these mutational profiles that really give us the answer of all of the possible sites that you can insert a domain within this protein that it tolerates and can become a switch, right? And so we take some of these blue ones that seem to still be active in this case and look at their function within this particular setting. And so we usually use this tool to build chemical sensors. And so one of the chemical sensors that we built is one for things that hit the human estrogen receptor. So broadly endocrine disrupting molecules, but the ones that specifically target the estrogen receptor. And so we took that domain, inserted it all throughout, found the hot spots where it seemed to still be functional, and then tested basically their response to molecules that trigger that receptor. And so the model molecule we're using here is 4-hydroxytamoxifen. This is a breast cancer drug. You do find it in some of the waste streams coming out of hospitals. It's not the most common environmental pollutant. But as you can see here, at low concentrations of this molecule, you get very little growth of these cells. At high concentrations, you get lots of growth. So this is showing you can turn on electron transfer and activate growth of these cells. And then you can just compare that to a wild type and an inactive version of these proteins down below. And so we've done this for 4-hydroxytamoxifen, but also a panel of other endocrine disrupting molecules. So you can take bisphenol A and it'll have a very similar response that you see here. And so we can take and we can build these types of control elements that can gate electron transfer. The next question is, can you actually plug them into the circuit and control the flow of electrons, right? And so the first thing we wanted to do was just test, can you measure the activity of a ferredoxin with one of these electronic interfaces? And so the pathway that I just described to you takes electrons from NADPH and makes sulfide out of them. And so we needed some way to couple this sulfide to this MTR pathway out on the cell surface. So some way to connect these to the quinone pool. Fortunately, there are tons of organisms that are sulfide oxidizing organisms that have sulfide quinone reductases that do this one enzymatic step. So we inserted one of these, and we built out an E. coli strain for expressing all of these components. So the ferredoxins, SQR, and then that output MTRA module, proteins they don't normally make. And then what we did is we wanted to test basically what is the current that comes out of these cells, right? And so we built a system where we had a non-functional ferredoxin, and we were testing if we add thiosulfate, one of the coupling agents here, what is the current that comes out of these cells. And so in the absence of a ferredoxin, you get the small amount of current you're seeing here. As you add thiosulfate, you don't get any change from this. And so without that ferredoxin being present, there's no connection for the thiosulfate to make it out to the MTR pathway. But then if you add in that ferredoxin, you get this nice sharp response. And so what you're seeing here now is electrons that are actually flowing through this whole pathway coming out to the electrode surface. And you can do this in a concentration dependent manner, so you can sense thiosulfate in rather quick time. And so what we wanted to do was then take our switch that we had built and see if this works within the same setting. And the way we normally do these types of measurements is to do ratiometric measurements. And so we'll have one electrode that's made of a control strain that doesn't respond to the molecule we're interested in, and then another electrode that has the sensing proteins within it, so that we can compare the current that's coming out of both of these. The reason why is to deal with changes in temperature or oxygen or other environmental conditions. We can actually back those out with this type of measurement. And so we went ahead and measured the relative signal that you get from the sensing strain compared to the control strain. If you use this molecule DMSO, which is our carrier here, you don't get much of a response from our sensing strain. But then if you add in that 4-hydroxytamoxifen, you get this sharp turn on electron current and signal coming out of these particular sensor molecules. And really the key innovation here is using these protein-based sensors. This is much faster than anything you would get with a gene expression-based sensor. And so if you actually go and quantify when we can detect this at significant levels, you're getting this in under 10-minute response times. That's really limited by diffusion of this molecule into the electrode surface where the cells are. So this shows that we can get these nice fast sensor responses. And so this was all done in laboratory media and nice clean bottles that these cells like to grow in, but we wanted to see if these sensors actually work out in real environmental settings. And so we went around Houston, actually Lynn, my colleague here, I think sampled the water for this particular example from these river bayou systems in Houston. You can see there's tons of organics in those, brought them back to the lab and tested if these sensors worked. And so we had some fresh water and marine ones. Again, you can see here, there's no real response to DMSO. And then when we spike in that molecule that triggers this receptor, you get a turn on and signal. So these sensors off the shelf and our sort of first proof of concept actually work in these much more complex environments than laboratory media. And so I just kind of wanted to wrap up and conclude on sort of the main takeaways here. I hope I've convinced you that we can now fabricate conductive living materials, that we can tune the conductivity of these materials. We could use those as sensors if we want, but if we want really fast sensors, we can actually engineer electron transfer proteins to be allosteric to respond to these molecules. And right now what my group's doing is taking that whole library of proteins I showed you that transport electrons, finding domains of interest and finding new sensors. And so we're thinking about PFAS, and it's a PFAS themed event. So we don't have a sensor yet, but maybe in the next time we talk, we will. Yeah. With that, I'd just like to acknowledge my budding group at Princeton, as well as all the people who were involved in the research before. Lynn, like I said, who's in the Aho Franklin lab, folks at Rice, folks at USC who helped with some of that optogenetic patterning. Happy to take questions.